Welcome everyone to the second PANGAIA seminar. Just to remind you, PANGAIA project is EU project number 872539, which is aimed to fill the gap between rapidly increasing powers of DNA sequencing and current limitation in resolving the inherent complexity of the data. So today's speaker is Tizian Schulz, who is a PhD student in the Genome Informatics Research Group at Bielefeld University, which is headed by Jens Stoje. He is also a student representative speaker of the graduate school Digital Infrastructure for the Life Sciences. Prior to joining this graduate school, he was affiliated with International Research Training Group Computational Methods for Diversity and Dynamics of Genomes, and also took a half-year internship in the Vancouver Prostate Center, hosted by Faraz Haag. And his research interests are mainly in the field of sequence analysis and comparative genomics. And today he is going to talk about searching for local similarity in Pangenome graphs. And with that, I will pass the work to Tizian.
Yeah, thanks for the invitation to present my work in this seminar series, and thanks for the introduction as well. Today I'm going to present some work about searching local similarity in Pangenome graphs, and this is joint work together with a couple of guys, Roland Wittler, my colleague from Bielefeld University, Sven Rahman from the University Hospital of Duisburg-Essen, Germany, Faraz Haag from Vancouver Prostate Center in Canada, and my supervisor, Jens Stoje, as well. Okay, so let me start with a short motivation. So we have a lot of DNA out there in our world, and nowadays we also finally have the technology to sequence all this DNA cheap and fast. In particular, we are able to sequence many individual genomes per species, and the number of sequences available in our public databases is constantly growing. So this introduces some challenges that we have to tackle. The first one is already the storage point. So considering that we have so many sequences that are actually highly redundant if they represent different individuals from the same species, for example, then it would be great to find efficient ways to store this data. Also, we would like to have efficient ways to analyze the data because we have so much of it, and we would like to do this analysis also as effective as possible. So we would like to get as most out of it as possible. So one way to tackle all these challenges is a pangenomic approach. In a pangenomic approach, we take all the sequences of some taxonomic unit, or for example, species, and we store it in one large object, which we then call the pangenome. And in the pangenome, we can make use of the redundancy of the data. We can store parts that are actually shared by all the genomes, or at least by parts of the genomes, only once. Well, then we only have to take care for storing all the unique parts that are unique to each of the genomes separately. And then also, Pangenome, we can jointly analyze all the genomes that are in this pangenome at once. And the data structure to actually that represents, or that may represent such a pangenome, is color-to-brown graph. In a chaotic ground graph, we have, well, considering some input sequences and some parameter k, we extract all the k-length subsequences of the input sequences, which we then call k-mers, these substrings, and we throw them in the graph. Each k-mer will be one node or vertex in the graph, and then there will be an edge between two nodes if their sequences share a k-1 overlap. Additionally, we have some color information that are stored together with each k-mer, and the color lets us refer back to the input sequences. So, for example, all the k-mers which have the red color, we know that they come from sequence S1, the red sequence. And now, if we traverse this graph, for example, by following a certain color, this allows us to reconstruct our input sequence. So if we go one step further, we can reduce the memory requirement of the quadratic ground graph a little bit more if we consider a compacted quadratic ground graph. In a compacted quadratic ground graph, all the nodes that are connected by a non-branching path, a single non-branching path, they can be merged, or they are merged into a single node, which we then call a unitic. And yeah, this reduces the number of nodes in our graph. And by this, it also reduces the memory footprint, and it speeds up the traversal of the graph. Okay, so why should we actually use such a graph to represent a pangenome? Well, there are a couple of reasons here. First one is we have a reduced memory requirement, because all the parts of the sequence that are actually shared by many individual genomes from our initial input set, they are stored only once, and only the differences we have to store separately. Then there's no data preprocessing necessary here. So in particular, we don't need any assembly. We can just take the raw sequencing reads from our sequencing technologies and extract all the k-mers, throw this into the graph, and we are done. There's no assembly step that we have to do. This is coming for free. Within the graph, the data is already stored based on similarity. What I mean is, if we look on the topology of the graph, we directly see which parts of the sequences are actually quite similar within all or a subset of the input sequences, and which parts are rather diverse. And of course, last but not least, if we analyze such a graph, then we can jointly analyze all the sequences that are represented by this graph. Okay, now the question is, how can we actually analyze such a graph? And before I come to that, we go one step back. We forget about the pangenomic approach, and we assume that we would store all the sequences in our in a sequence database. Then there's a famous tool that I guess everyone listening knows already. This is the basic local alignment search tool, plast, which allows to search a query sequence or search for alignments between a query sequence and sequences stored in a database. These sequences might either be DNA sequences or protein sequences, and we can even search for mixed things. So we can, for example, have a DNA query translated into a protein sequence and then search within a protein database. Then, what all these different flavors of BLAST have in common are three basic algorithmic steps. So we have first the first step where we try to find short similar substrings between the query sequence and the sequences in the database, and we call these substrings BLAST hits. And then these hits are extend it in a second step using an X-drop algorithm, as it's called. And then, last but not least, we take the best findings from the second step and we do a gapped recalculation of them. In more theoretical terms, what BLAST is doing is that it has a query sequence and a sequence and a database of sequences, and it tries to find all highest scoring local alignments between the query and the sequences stored in the database, which are above a certain significance value. Now, what our idea was, well, can we actually just do the same for a graph that stores all our sequences? And yeah, we developed a method that is able to do this, to query a color-to-brown graph in a BLAST-like manner, and we call our method Pangenome Local Alignment Search Tool, or PLAST in short. The theoretical problem that we are trying to solve here is very similar to what BLAST does. The only thing that is different here is that we represent all our sequences not as linear sequences in the database, but as a color-to-brown graph. If we look on the overall procedure of the algorithm, then this is also very similar compared to BLAST at the first glance. So we have a seed detection step as the first step, where we try to find exact matches between the query sequence and the graph sequence. Then in the second step, we try to extend all these seeds without gaps. And then in the third step, we do a gapped recalculation of the best findings from the second step. Okay, now I'd like to show you how this works in detail for some example. So let's assume we have this compacted color-to-brown graph here, k equals 4 in this case. Then to understand the algorithm, the basic algorithm, we can first do some simplifications. First of all, we can forget about all the color information, because for the basic algorithm, this color information is not necessary at all. Also, we can ignore the edges for now, because I mean, in a compacted color-to-brown graph, the edges are not explicitly present anyways. They are just defined by the overlaps of the sequences of the nodes, so we can also ignore them for now. Then we need some indexing of our graph for the first step of the algorithm. For this, we choose a minimal exact match length, which we call S, and then we calculate an index. The index consists of 2 arrays. In our case, the first array is a S-merge profile, which stores all the number of, for each possible S-merge, stores the number of occurrences of that S-merge in the graph. And then the second array stores the actual locations of an S-merge in the graph. This is done by a pair, so we have a pair of vertex ID, so the vertex where the S-merge appears, and then the offset in the sequence, at which position in the sequence the S-merge appears. And then the first array we can use to find the corresponding entries in the second array, and from the second array we can find the S-merges in the graph. How this works in detail, I'm going to skip here for time reasons. Okay, so now first step, we want to detect seeds. We have a query now down here, and we can find a seed TAACG in the middle of this query sequence. So there are much more seeds that we could find here, but for our example, let's just focus on this one seed. Now this first step is kind of straightforward. It's actually the same what BLAST does. So it's not conceptually not much of a difference here. We can do the same even if we consider a graph instead of a sequence database. Then, in the second step, we would now want to try to extend this seed to the left and to the right. And for this, we compare the query sequence to the sequence in the graph. So first here we consider, in this example, we consider unit scores. So the first base that we are comparing here is a mismatch, G and T. Then we get a match, and then we have reached the end of the unitic sequence here. And now the question is, what can we do? Because the graph at this point, it branches off. So this means that there are different possibilities how the graph sequence could go on. And we have the task to find the one graph sequence best matches our query sequence in order to get the best alignment. So how should we do this? Well, we do a depth-first search on the successive unitics in the graph. So now for this example, we just continue with this unitic here. We get another match if we compare the sequence. And at this point, this is the first time when our overall extension, the black part, has an overall positive score, a score of +1. And this means that if we would choose this extension, we would already have increased the complete score of our alignment, also including the seed. And this is why we would like to, well, first of all, mark this. So keep it in mind. This is a possible extension that is good. And then we go on this unitic here. However, we cannot find anything further here. So we are done on this. On this unitic, the extract algorithm that we do use as well drops at this point. And then we are back at our outgoing unitic, and we try to find a different path through the graph. So we go to the unitic up here, and again we do some string matching. And at some point, we again find an extension that has an overall positive score. And we compare it to what we have already found and keep the best. And then we go on with our extension. In this case, again, we cannot find anything more. And now we would have to do the same to the left side of the seed as well. In our example, however, we cannot find anything to the left. So as you have seen, this is more expensive than what BLAST can do on a sequence database, because we have this ambiguity in the graph sequence. There are different possibilities how to traverse the graph that gives us different graph sequences. And we try, with using a depth-first search, we really try every possibility how the sequence could go on. This is a lot of work. However, this is mostly feasible. So as long as the graph does not become too complex, this is feasible. And most of the time it does not become too complex. However, there are, or there might be parts in the graph where the graph is actually too complex, so there might be collapsed regions in the graph. These regions have many unitics with very short sequences, and they have many short loops, and the vertices have a high branching factor in general, which means there are many different possibilities to generate a graph sequence here. And actually there are even so many possibilities sometimes that it's almost possible to find any possible or almost every possible continuation of our graph sequence. So there will be like almost always something that somewhat matches our query sequence. So we will get through this region quite fast because there is some path that matches somewhat our query sequence. But then we have to go back. We have to try all the other different possibilities as well. And this is where the approach starts to become inefficient, because there are simply too much possibilities here. And we cannot gain any further information because we will definitely find more paths that are somewhat matching the query. But to find the best one, we just have to try everything out. But this is not worth the effort in this case. We can also stay with something that might be suboptimal, but that's good enough here. And how we deal with this is, well, we introduce a threshold. We count the number of switches between uniticks during an extension, and as soon as this number of switches exceeds the threshold, we just stop the extension at that point and keep what we had found so far. Okay, this solves also the problem of the extension for the collapsed regions in the graph, and now we are left with the third step of our algorithm, which is the gapped recalculation. And this is even more expensive than the second step, because we have to calculate a banded alignment to consider gaps for every unitic in our graph where our path guides us. And but this even though this is more expensive, it's not a problem in our case, because we only have to do this for a small subset of all the findings from the second step. The second step, however, we have to do for all seeds that we have found. We don't know which of the seeds will give us a good alignment and which one won't give us a good alignment. So we have to do the second step for all the seeds. The third step we only have to do for a small subset of all results, and this is why this is not not too common, not too much a problem time-wise. Okay, so that's the basic algorithm already. So what can we do with this? Well, this algorithm will find us highest scoring alignments between the graph sequence and the query. What we have to keep in mind here, if we use default parameters, if we use the algorithm with default parameters, we haven't made use of any color information, which means that the result does also not have to be consistent with any input sequence. So if we have found an alignment between the query and the graph sequence, it does not mean that that the sequence of the graph that is aligned to the query in that alignment represents any of the input sequences. It can also be that the beginning of the alignment represents a sequence that have appeared in the red genome, for example, and the end of the alignment might have appeared in the blue genome. We have to be aware of this. And now we can make use of the colors as well, and we can do this, for example, by using a quorum. A quorum is a number, just an integer that we can use to focus to limit our search on parts of the graph that are covered or supported by a certain number of individual genomes. So if we, for example, only want to search for alignments which are covered by at least 90% of all the input genomes of our pangenome, we can use this quorum parameter. Other than that, we can also make use of a search color set. A search color set is a set of colors that we define as input to our algorithm, and set limits the search on path or parts of the graph that are actually covered by the sequences of the search color set. And of course, we could also use both the quorum and the search color set in combination to make even more precise searches in the graph. Okay, so now we are almost done with the algorithm. There's only one important step missing, which is alignment statistics. So what plast does is that it calculates an E value and a P value for all the alignments that it finds to estimate the biological relevance of the findings. So the question here is, well, the alignment that we have found, does it actually represent homology between the query sequence and the graph sequence and the database sequence? Yeah, and to determine this, there are the e-value and the p-value, which are calculated here to get an estimate. What we calculate here is more or less the probability to find the same alignment in a random context. So if there's no homology at all. The p-value is dependent on the e-value, so I will only focus on the e-value here. And the e-value is calculated in the following way. We have some constants, lambda and k, and then there is the length of the query and the length or the size of the database, which influence the e-value in a linear way. And then we have the score of our found alignment, and the probability to find an alignment with some score decreases exponentially if the score increases. So now the question is, does this apply on a pangenome, on the pangenomic context where we use a graph to store all the sequences? Well, here we have some assumptions. So the first assumption is that the sequences in our graph are highly similar. This is because we are considering a pangenomic context. All the sequences come from the same taxonomic unit. There is a lot of similarity between these sequences, and this is one thing that is different compared to the BLAST statistics, because BLAST assumes that all the sequences in the database are independent from each other. So this fact that the sequences are similar here should actually reduce the probability to find a random alignment between the query and the graph sequence. On the other hand, however, we have this branching in the graph. The more branching there is in the graph, the more possibilities we have to generate a graph sequence, and even further, we can generate completely new sequences that we have never inserted into the graph, and this should definitely increase the probability of a random hit here. However, what should stay the same is the assumption that our maximum score should follow an exponential distribution. This is what we assume. And then our statistical parameters, lambda and c, they depend on the relatedness of the sequences. This is the first point here. And they depend on the diversity of the sequences. This is the second, the branching. But the details are unfortunately unknown. We had a look into that, but we couldn't find any, say, easy rules here. There is, nothing too easy to deduce here. So we cannot make any further assumptions. And in order to determine our statistical parameters, we used the following approach. We did simulations. So we estimated the underlying probability distribution by generating random queries and calculated alignments between the random queries and the graph. And with this, we try to estimate the statistical parameters lambda and c. So now there's a problem with this random sampling, which is that if we generate a random query, this query will usually get a very low score if we align it to the graph. But because we actually want to estimate our parameters for, the parameters should be especially good for the regions where we actually want to calculate e-values for, and these are usually a little bit higher scores. So what does this mean? Well, we would actually have to sample a lot, do a lot of query generation and scoring here in order to get reliable parameters for this score regions. And this takes an incredible amount of time. So what we did instead was we used an importance sampling approach. And yeah, what is the idea here? Maybe just shortly in a nutshell. So we generated or we created a Markov chain which allowed us to generate random query sequences which were a little bit more similar to the graph as they would be if we would use just the naive sampling, let's say. So in particular, we use this Markov chain to exponentially bias our queries towards higher scores. How this works in detail is described here, but I'm going to skip this. Just to have an idea, so we have this Markov chain, it generates us queries which are exponentially biased towards higher scores. What we can do then is calculate scores for these queries against the graph and count the number of scores that we, or the number of how often we observe a certain score when doing this. And then if we have this, then we can even out our introduced bias again, and we end up with some data points which we can use. If we plot them in a semi-logarithmic space, then we will be able to fit a line on this point, and this line will allow us to estimate our statistical parameters, lambda and c. Yeah, so this is maybe a little bit abstract, I guess. So let's have a look on some pictures to make this more clear. So here on the left, you can see so to say, the probability distributions that we could estimate by our importance sampling and by our naive sampling. So we did this for ungapped alignments and for gapped alignments. And as you can see, if we use gapped alignments, the score is in general a little bit higher than as if we would use no gaps. That was to be expected. And then both the gapped and the ungapped alignments we generated in two ways. On the one hand, we used the naive sampling approach. This is the crosses in this plot. But it was only possible to get them up to a certain point because, to get to higher scores would just take too much time. And then we also used the importance sampling approach, which is the pluses in this plot. As you can see, the pluses very nicely allow us to elongate these distributions towards larger scores. So that worked quite well. And then we can use this points here to fit a line. And the slope of this line will be our lambda parameter, and the intercept will be our c value. Here on the right, you can also see the histograms of the scores that we sampled here. On the one hand, using the naive sampling, we only find a very small range of scores, but these scores we actually find very often. And then if we use the importance sampling approach, then this allows us to sample from a much larger range of scores. And we can sample from this range almost uniformly, not completely, but at least we find all these scores like approximately with the same abundance. Okay. Okay, so this was really the whole algorithm. This is how it works. Now let's compare it to BLAST. So what does BLAST do? It compares a query sequence against all the sequences in a database. And this is what we can do in a similar way for the graph. So we can compare our query sequence to the graph sequences. So the sequences that we can generate with our graph. But we can do more. We can also compare all the sequences in our graph among each other with respect to the query. What do I mean with this? Well, if we have an alignment, if we have found an alignment, and we have a look on the colors that appear in the graph sequence that is involved in this alignment, then from looking on this, we can see which sequences from the graph are actually very similar. This is because they appear together in the alignment, and which are not so similar because they, for example, do not appear together in the alignment. Then what BLAST does is that it processes every sequence in the database independently, while we can actually process many sequences jointly if they share the same path in the graph. And if the sequence similarity increases, and this is the case in a pangenomic scenario, then the runtime of BLAST will increase because it will have to spend a lot of time for every single sequence in the database if they are all similar to the query. And for us, this is actually the other way around. The more similar our sequences are, the less complex the graph will be, and the more time we can save here. Okay, so far in theory, but is the practice actually agreeing to this? This we try to find out with this experiment. We had a pangenome of 5,000 Salmonella strains, and we used 100 random queries drawn from the Salmonella reference genome to search for them in the graph. If in the pangenome. The graph is we only search in the graph in the case of plast, but not for the other tools. So then we built subsets of the whole pangenome of different sizes up to the whole pangenome size. And this is what came out of it. And we can see that the tools that we compared here, some of them they took very long. Some took that long, but still longer than us. And there was only basically one other tool, this MMSEC2, which was able to compete with plast in this experiment. And what I would say you can also see here, you might agree or not, is that compared or in contrast to all the other tools, plast's runtime grows sublinearly if we increase the number of genomes in our pangenome. But well, you might also say we would have need more data here to really see this. What we can maybe agree at least is that plast is among the fastest tools in this experiment. So in this plot, MMSEC2 was kind of equally good compared to plast. Let's have a look on the memory. And here it is. This is not true anymore. If we look on MMSEC2, it does not appear in this zoomed plot at all, because even for the smallest pangenome size, it uses already 9 gigabytes of main memory, and then this requirement grows enormously, very fast. Plast, however, is able to use a lot less memory. And this is mainly because we are using a graph, which allows us to exploit this higher redundancy between the sequences. Okay, so then we also did another experiment, and here we were actually trying to simulate a possible use case of plast. And for this, we used a study where a pangenome of 21 Vibrio cholerae genomes or strains was taken. And the question of this study was, okay, so what actually makes this, I mean, there were some strains which were taken from the environment, and there were some strains taken from hospital patients. So there were some pandemic strains, some pathogenic strains, and some non-pathogenic strains. And in the study, they wanted to find out what actually makes the what they call the pandemic genomes, what makes them pathogenic. And they found out, well, there is this region, VPI1, consisting of 19 genes that cause virulence. And this is why the strains taken from the hospital are pathogenic compared to the others. So now we wanted to know, can we actually validate this finding using plast? And for this, we used the whole region as a query for our algorithm, and we built a pangenome graph of this 21 genomes, and then we searched for alignments. And what we found is visualized in this left plot here. So you can see that we could find something for the whole, like alignments for the whole. This is not one continuous alignments, because in some genomes also the order of these genes is not conserved. So we found, but we found alignments that actually covered the whole query. And the colors that we found for these alignments were these colors here, and all these colors, all these strains are they belong to the pandemic, or they form the pandemic group. So that there was basically no color present of the environmental genomes. So we were wondering, okay, where are these environmental genomes? And to find this out, we made a second search, and this time we used these genomes as a search color set. And now only alignments were outputted where these colors actually appear. So we excluded these alignments here. And what we could find is shown here on the right. So we almost find nothing. Sometimes, especially for these two genes and here in the beginning, we found something, but in general there was a lot of nothing for most of the environmental genomes. So that was quite an impressive result, I found. And this was possible only with two searches. So one search with plast without a color set, and one with a color set. I don't know how much effort it was for the research study to do this. Okay. Then there is a last question that I would like to, for which I would like to present you in an experiment here. So we ask ourselves the question: okay, can we actually also work on pangenomes of genomes of mammalian sizes? And for this, we constructed a human pangenome. So we used the variant information from the 1000 Genomes project, and we used the human reference genome, and we reconstructed all the 2,504 human individuals. Or to be more precise, we reconstructed chromosome 2 of all these 2,504 individuals. and yeah, we formed a pangenome out of this. Yeah, and then we made another performance analysis using this pangenome, the human pangenome, against MMSEC2, because this one was the best performing tool from the previous experiment. And yeah, what can we see? Well, plast took on average for every query, like we again used 100 queries. This time we drew them randomly from the reference, from the human reference genome. Yeah, and plast took like on average 317 seconds for this, and used 24 gigabytes of main memory and a single core. MMSEC2, in comparison, took only for a subset of 1,000 chromosomes, so not the complete 2,500, but only 1,000 chromosomes. It took already 339 seconds on average, using 259 gigabytes of main memory and all 28 cores that were available. And also, if we look on the disk space, yeah, we can see something similar with regard to the memory. So plast used for the graph and the index used in total 9.2 gigabytes. And in comparison to that, MMSEC2 used for the sequences and the index 2.2 terabytes of disk space. Okay, so to come to an end, let me give you some conclusions. So I hope I could convince you that the growing amounts of available DNA sequences motivate the usage of pangenomic approaches. We have seen that querying a colored Brown graph is actually possible in a blast-like manner. And yeah, if we do this comparison, then this allows us not only to compare the query sequence against the graph, but also to compare the sequences within the graph, yeah, jointly. Okay, so if you are interested in more details, here's a link to a preprint of our work. And yeah, thank you for your attention, and I'm happy to take questions.
Thank you very much. I will remind you that you can ask questions here, or you can ask questions on Slido. So, are there any questions? So there is, yeah, Travis, please go ahead.
I'll just remember to lower my hand. I was wondering, so could you sort of do a mix of blast and plast, where at once, so in plast you have to when you find the seed, it may not actually be consistent with any of the colors. Whereas, so if you sort of search for the seeds in blast, you could then, so once you find the seeds in blast, you could then swap it over to plast and say, okay, how do I extend that seed in the Bruijn graph?
Yes, and then, like also during the extension, you would want to try to stay consistent with at least one of the input sequences.
Yes.
Yeah. I guess this is possible. I even already thought about how you could actually implement this into plast. So this is definitely the thing that is possible. So we can also, during the extension phase in plast, we could yeah, take care of the colors and to take care of staying consistent with at least one of the colors.
So there is one question on Slido. The question is, have you tested plast on any plant genomes? I guess these are much longer than the mammalian genomes, or.
Yeah, that's true. We haven't done this yet. to be honest, but that's a good point. We should try that out. Currently, I'm working actually on an experiment that considers the whole human genome. So not only chromosome 2 as in this experiment, but the whole human genome. And yeah, I guess I'm very confident that this will work out. I don't have any final results, so I won't say anything about this yet, but I hope that it will work. I'm actually quite confident, and then also it would make sense to try it out on a plant genome, yes.
It would be also great if you can switch on your cameras during the discussion. And Bruijn, please.
Hello, so I wanted to ask, as far as I understand, your seed length S is less than K used for building the graph. What if you wanted to use longer seeds, maybe some minimizers or something like that?
Well, I mean, in this example, K was very small, right? So usually it's like in the range of, I guess the default for the underlying data structure that we use is 31. And so the seed length could be like everything up to 31. And but a seed length of 31 is already quite long usually. Like BLAST is using, like nucleotide BLAST is using, I guess, a seed length of 11 by default. So yeah, I guess we would lose a lot of sensitivity here if we would even use longer seeds than 30 or 31. So this is not. I mean, this has not been yet a focus on.
Yeah, but if you use, for example, minimizers, you look at, instead of just simple seeds, you would look at a longer window and within that look for...
Okay.
So then maybe you could use the 31 to use minimizers with shorter seed length.
Yeah, that's an interesting idea. Actually, the data structure that we use, Bifrost, this data structure uses minimizers anyway, so only one could make use of that. Yeah.
But wouldn't the longer windows, I think from what you have mentioned, it seems that the seeds don't go across the boundaries of the nodes in the graph, right? So,
yeah,
so if there were longer, sort of windows, then you probably would run into problems with that.
Yeah, well, this could cause a problem. Yeah, I mean, I would have to think about it. Yeah, for a longer time. This could definitely make problems, and this is why so far we stayed on seeds that were shorter than K. Yeah.
And perhaps strategies like multiple seeds and things like that have proven efficient with the BLAST.
Yeah, like this two seed approach that BLAST uses, where you need two seeds that are sufficiently close to each other to actually start an extension at all. Yes, that would also be a possibility. The problem, however, here, I mean, as soon as we have a graph, the problem is that it can be a lot of work to actually estimate the distance between two seeds if they do not lie on the same unitic. Then yeah, we again have to traverse the graph, and this can take a lot of time. And since the seeding step is like the most crucial for the overall runtime, because we find so many seeds, we have to be very careful with this.
Okay, Sandeep, please.
Hi. Yeah, I was the one who asked a question on Slido about the plant genomes. So I was wondering if plast is available for checking out on GitHub or any such code share.
Yeah, so we have it available on our GitLab, so our university GitLab, where you can. This is like very similar to GitHub, so you can just download it from there. Yeah.
Awesome. I'll try to test it on some rice genome project that we have. We're working within the Pangenomic project, and maybe I'll talk to you frequently about the results that I get and I need your help.
Yeah, that would be great. Yeah.
Interesting tool. Thank you very much.
Thank you.
So, any other questions? So I was actually interested in, So you were mentioning that you used sort of a heuristic or something like that for like to avoid too many branches. So basically, you, as far as I understand, you penalize for switching unitic. And wouldn't it be more appropriate, perhaps, to penalize for switching colors, especially in the complex regions? Or would it be more difficult in any way?
It would be more difficult because in the implementation of the color-to-brown graph that we use, it can be that the color set changes within a unitic. So we would not only have to check the colors at the end of every unitic, but we would also have to check it for every position within a unitic. And as the color information is compressed and it takes quite a lot of time to access this information, it will probably slow down the whole process. Regarding the collapsed regions, I was already thinking about some kind of masking approach. So what we know from BLAST as well is that you can just mask some parts of the sequence to avoid that they cause problems during search. So this probably would also be an approach that might make sense for BLAST. The only thing that is missing here is that I have to think about a way to identify these regions in order to be able to mask them properly.
So, any other questions? Shira?
Yeah, hi. Thanks, Tizian, for the talk. I was wondering, so I have two questions actually. One, so first is, there are many sequence to graph aligners out there, like mini-graph, graph aligner, and to my knowledge, all of them are actually solving, or at least attempting to solve local alignment problem, because they are all looking for local matches. So I was wondering that have you attempted to compare BLAST against any of the recent sequence to graph aligners?
So as far as I know, actually these methods do read alignment, and in read alignment you can also follow some seeding strategies, but what you try to calculate is not a local alignment but a semi-global alignment, and especially for graph aligner. I know this because I was in contact with the authors as well. This is not possible because for a local alignment you need a score system. So you have to calculate an alignment score. You are searching for similarity, but not for difference. So you don't have a cost scenario. And graph aligner, for example, only works for a cost scenario.
Okay, yeah, thanks.
Any other questions? I actually have one more. One of the uses of BLAST is that you sort of take, or similar tools, is that you take two big genomes, let's say, and you find all similarities between these genomes. Were you thinking about a problem where you would take sort of two Pangenomes and find all local similarities between two Pangenomes instead of having a string query and the graph on the other side?
Yeah, so this would mean like it would basically generalize the idea of a genome alignment to Pangenomes, so do a kind of Pangenome alignment, right? Yeah, well, to be honest, I mean, I thought about it maybe, but only very very roughly, and I guess there's a lot of more problems approaching if we want to try to calculate alignments between two graphs actually. So I guess this is much beyond this work that I did so far. I guess maybe it's possible, but so far I hadn't had a good idea how to do this. you.